Okay, it's 5:30. So how are you all?
Good, Miss Nandi. How are you?
Feeling sleepy?
Yep, same here.
Yes, rainy weather is a thing.
Yeah. And I got stuck in
No.
traffic. Yes, I just came back.
Wow.
Oh, yeah. So, okay, so I think it's already 5:30. So let's start. Yeah, it's 5:31. So let me start. Um, um, so today I'm going to start chapter 6. Okay, and this chapter talks about acellular pathogens. And I hope you remember that. What does it mean? Acellular means they don't have any cells, right? So basically, we are going to talk about virus type of microorganism in this chapter. So this chapter actually will talk about structure, the composition of acellular pathogen. Then it will, just a minute. Then it will talk about the classification, so and cultivation. So basically we are going to work on the structure, classification, and cultivation, how you culture. There are special procedure because the virus or all acellular pathogens, they are so small, and because they are not cell, they need a special stains, a special microscopes and all those. Okay, so it's very difficult to cultivate them in lab. They need host cells. So mostly we are going to work on the viruses this chapter, and then there are a few other acellular pathogen, we are going to, we will just touch about them, just the definitions and very little bit of characteristics and a few diseases they are responsible for. So we will study in detail the virus, and the rest of the pathogens we will just touch actually. So who are the acellular pathogen? Okay. Um, the bacteria, we actually studied about that bacteria until the microscope discovering of microscope. We really did not have any idea about the bacteria, right? um, Acellular pathogen, the people, they have an idea, they were aware of some type of disease that is not coming from the bacteria, but they did not have any idea um, about an organism which is much smaller than the bacteria. Okay, so until uh, the discovery of electron microscope, we really couldn't find out anything about the virus. So that's the electron microscope. So you can understand that our regular light microscope or bright field microscope, that which one we use in lab, those are not capable enough to show us or to enlarge in such a way so we can see the virus. So it's not. So we need electron microscope. Electron microscope is really costly. There are few research lab where they do have electron microscope. So this acellular pathogen, who are those? They are the viruses, viroids, virusoids, and prions. Virus and prions, virus actually, they are for animal, they are the pathogen for animal, they are the pathogen of bacteria, they are the pathogen for the plant. Okay, prions, these prions, they are pathogen of animals. Okay, these two, viroids and virusoids, these two are pathogenic for the plants only. Okay, now these viruses, as I told, that they are acellular organism, means they really do not have any cell. Okay, what they do have, they have only protein and nucleic acids. Nucleic acid means either DNA or RNA. Inside a virus, you cannot see both DNA and RNA. Okay, DNA, either DNA or RNA. Okay, just one type of nucleic acid they have. So you can understand that just they don't have cell membrane, they don't have cell wall. Only thing they have, protein and either DNA or RNA. Viroids, they only have RNA. Okay, and that's also single-stranded RNA. Virusoids, they have single-stranded RNA, but they're so minute um, that they and as I told, that they are the pathogen for plants, but they cannot attack plants until that plant is already infected by the viroids. Okay, or virus. If they are not infected by the viroids or viruses, then virusoids, they are inactive, they cannot make a plant diseased. So that's the virusoid. So virusoids, to give the disease or to multiply, that plant has to be already infected with viruses, most of the time with viruses or the viroids. Otherwise, they cannot infect the plant. and viroids and virusoids, they only have single stranded RNA. They don't have anything else. So you can understand, like just imagine that one DNA that can make a plant sick, or that can make us sick. Okay, so that's the viroids or virusoids, and that also single stranded RNA. Prions, that is actually a piece of protein, very small piece of protein, and that protein, we all carry that protein, we do have that protein. Okay, for all of us, that protein is inside our body, but suddenly that protein get mutated. One type of mutation occurs, and that protein converts into prions, okay, or prions, and that protein can give you so many dangerous disease. So they are very pathogenic, that protein, when it gets mutated. So these are, at this moment, these are the 4 types of acellular pathogens we know, okay, at this moment. In this chapter, we are going to work in detail the viruses, and these 3, viroids, virusoids, and prions, we will just touch very little. We will study. So the virus, okay, viruses were too small to be seen with the first microscopes. Okay, we needed electron microscopes. So until electron microscope has discovered, we couldn't see the virus. We really did not have any idea. Louis Pasteur first proposed the word, the virus. He gave the name. Pasteur and another scientist, his name is Chamberland, or you can say Chamberlain, whatever. Pasteur and Chamberlain together, both of them, they're in Paris that time. So together, they actually make one special type of filtering equipment. So it was made up with porcelain. Okay, so porcelain filtering equipment, you can say. So they made for their own purposes, but it was the joint efforts of Pasteur and Chamberlain. But they didn't use this filtering membrane to observe the virus. They just made the filter machine or equipment. Ivanovsky, okay, he actually used, so that time what happened, that all the scientists were really busy to discover that tobacco mosaic plant, okay, tobacco mosaic virus for the tobacco plant. Okay, they did not have any idea about that virus, but one type of disease that was actually destroying the tobacco industry. So a huge amount of loss. So lots of researches were going on that time. So Ivanovsky, what he did, he used this filtering equipment. That filter paper, the hole, or you can say that slits were less than 0.02 nanometers. So you can understand it's really a minute hole. Okay, so bacteria cannot pass through this hole. So, but they made, Pasteur and Chamberlain made it for their own research purpose. So Ivanovsky, what he did, he used that filtering paper to filter that tobacco mosaic leaf to see that if it is a bacteria or what it is, to see that. So he found out that it does not looks like bacteria, but something is there who are really, really minute. So he told maybe it's a very small, some type of bacteria. That's what he thought. Okay, but he was the first person to show the world the actual very small, minute microorganism. He was the first person. So that's the reason Ivanovsky is credited as the discoverer of viruses, and he is called the founder or the father figure of the field of virology. Study of virus is called virology. Okay, so study of virus. So that's the reason, but he couldn't discover, he didn't tell that that's the virus or completely separate microorganism, but he thought that's a bacteria, but it's a very small size, much smaller than the bacteria. Then Bazarnik, what he did, he actually he did the same experiment what Ivanovsky did. He discovered that that's not the bacteria. He thought that that's a kind of toxic chemicals. That's what Bazarnik thought. Okay. Um, so uh, he didn't get the credit, but Ivanovsky got the credit. But we had an idea this disease that that was infecting the tobacco mosaic plant, that was actually not bacterial infection. That we got, that idea we got from Bazarnik. Okay, so that's the history of the virus. Why can't I? Okay. So now the characteristics of virus: infectious, acellular pathogen. Why they are acellular? Cannot exist independently from the host cell, so they are not considered as living things. Okay. Now I hope you remember that what's the difference between the living and non-living material. Okay, so to be living organism, it must have few characteristics, right? So it must have capability of doing metabolism. It can grow, and so you can say the growth and development. That's another important character to be considered as living. It can reproduce, so reproduction. Then sensitivity. Okay, that another one. Most importantly, all the living cells are made up with a cell. Okay, it could be one cell or multiple cell, but it must have cell. So there are a few more. So anyway, so these are very important characteristics to be a living organism. Okay, these viruses, they don't show any of these characteristics. They don't have metabolic capability. They cannot grow. They cannot develop. They can do reproduction. I'm coming at this point in shortly. Sensitivity they don't have, and they are not made up with cell. Okay, now the reproduction, yes, they can do reproduction, but when they are inside a host cell, inside a living cell, then only they can do reproduction. Otherwise they cannot. When they are in the air, they're in the environment outside of a living cell, they cannot reproduce, they cannot multiply. Okay, so basically if we see that these organisms, they really do not show any characteristics that represents their liveliness. Okay, so we actually cannot consider them as living organism. Okay, But at the same time, we cannot consider them as non-living organism, because when they are inside a living cell, they can reproduce. That's the only one characteristics. Because of that virus, they are in between the living and non-living organism. We are not sure. Okay, so that's the virus. Acellular because they don't have cell. Um, Reproduction, as I told, they can do only when they are inside a living cell. They are actually obligate intracellular parasites. Obligate means must, intracellular parasites. They behave like a parasite. They are pathogenic. They can make us sick only when they are inside a cell. Okay, so to be parasite or to be pathogen, they have to be inside a cell. So that's why they are called obligate intracellular parasites. Okay. So they are obligate intracellular parasites with host and cell type. They have tissue tropism. Not only that, they have very specific, it's called host range. Okay. So basically what happened, these viruses, they are very, very picky about the host. Why? Picky means basically, when, let's say this is one virus, okay? This is a, let's say this is a virus. Now, and this is one animal cell. Okay, so let's say this is SARS-CoV-19, CoV-2 actually virus. Okay, outside of this virus, there must be some type of glycoprotein. One special type of protein should be there, and I hope you remember that we also have special type of glycoprotein outside of our cell. That's our ID tag. Okay, so this is called receptor, and this is called ligand. So this protein has to match with that protein. If these two matches, then only virus can sit and can go inside one animal cells or any living cells. Okay, so that's the main criteria, and that's the reason they are very, 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 very host specific. Host specific, or the word host range means, like if one virus, it can infect bacteria, that virus won't be able to infect animal or plant. Okay, and that's the reason we actually classify virus like that way, like bacterial virus, okay, or maybe the animal virus or the plant virus. So all the viruses, they're very, very specific about their host. The plant virus, they cannot infect any animal. Animal virus cannot infect plant or bacteria. Bacterial virus, they cannot infect animal or the plant, because of the special type of protein or receptor they get this host range. Okay, so that's very, very specific and special type of characters for the viruses. Okay, now they have tissue tropism. Not only they are picky about the host, but they're picky about the very special type of cell, or you can say tissue, like this new virus like SARS. It's called SARS-CoV-2. This is the virus. We are going through it now. This virus, they only can give the infection if they can enter through the upper respiratory tract. Okay, so those are the tissue they have receptor for. Okay, or eye conjunctiva, your conjunctiva, that's the the outside of the outermost layer, you can say the clear transparent membrane, thin membrane, that's the conjunctiva. Okay, so that conjunctiva and upper respiratory tract, all to the larynx. Sometimes some scientists consider larynx as lower respiratory. So basically your nose, okay, then pharynx, and little portion of larynx, that's the upper respiratory tract. This area has that receptor for that virus that actually matches. Okay, so um, if the virus goes inside your mouth, digestive tract, esophagus, let's say, with your food, they won't survive because they don't have receptor over there. Okay, they cannot even sit onto your cell. Okay, but that upper respiratory tract and eye, not only the conjunctiva. Now that day I was reading that upper eyelid also they have receptor. Yeah, you can say that upper eyelids. And that's the reason, without washing your hand, you really should not touch your nose, your eyes. Okay, so that specific characteristic is called tissue tropism. Okay, flu virus, all virus has this very um, strong affinity with a special type of tissue, and that is called the tissue or cell, that is called the tissue tropism. Flu virus, influenza virus, if you inhale, if you take it with food, if it goes through your esophagus digestive system, you are not going to get that influenza. But if it enters through the upper respiratory tract, then you are going to get it. So that's the affinity of influenza virus. Okay, so affinity with the upper respiratory cells, basically. Hepatitis virus. Hepatitis virus, they have affinity for liver cells. They only can sit or only can attach with the liver cell. Okay, so if you have hepatitis, then your liver is going to infected. Polio, nervous system, your brain and spinal cord for the polio. So lots of example I can give, but so that is so attraction for attraction to a very special type of cell or group of cell, means the tissue, that is called the tissue tropism. And all virus shows this host strength and the tissue tropism. Very interesting characteristics. They have a genome, either DNA or RNA. This thing is very important. Never both, either DNA or RNA. Now DNA and RNA is surrounded by a protein capsid. Sometimes they do have a membrane, and the glycoprotein cannot multiply unless they invade a specific host cell and instruct its genetic and metabolic machinery to make and release new viruses. They are like what they do, they are like lunar lander. Okay, on any, you can say, any alien planets. Okay, they just land and ready to unload all the genetic material. That's how they behave. So what they actually do, you can understand that they have little bit of protein and DNA or RNA. Once they get attached with your cell, they enter. They have only DNA or RNA. what they do, and the protein means they have very few enzyme. They take control over your cell. First thing what they do, they actually inactivate your chromosome. So your all the genetic material, they cannot do anything. And then they take control over the cell and they direct host cell to make more copies of that virus. Okay, that's how they multiply. And most of the time, not most of the time, they use all the enzymes, proteins from your body, from host cells. Okay, all the machinery, like to make protein they need ribosomes, right? They don't have ribosomes, so they use our ribosomes. So that's how they actually multiply. They force our cell to copy all the DNA, means viral DNA or viral protein, and that's how they reproduce. And for that they must have a living cell, otherwise they cannot do that. So as I told, that host range, they have very specific host range. That virus which actually infects bacteria, they have a special name. We call those as bacteriophage or phage virus. That's what we call them. Okay, so bacteriophages for the bacteria, animal viruses, and the plant viruses. These are the three main types of virus based on the host. Now the transmission. Okay, so if we talk about, let me see, I really want to I really want to get a blank board. I cannot. Okay, so so the transmission. Okay, so transmission, let me write it over here. So two different type of transmission, means how they transmit, how we get them. Okay, so one is direct transmission, another one is indirect transmission. So when it is direct transmission, example like flu virus, that's a direct. Okay, COVID-19, that's also direct transmission. In this case, what happened? The person, let's say it's sneezing or coughing, or sneezing, coughing, and you catch the airdrops. Okay, and you can get them. So that's the direct contact, direct transmission. So not the contact, direct transmission. You are too close to that person and you can inhale whatever that person is releasing, and with those airdrops you can actually inhale the viruses. So that's the direct transmission, and these are the examples. Indirect transmission, two different types. One is by the fomites and another one is by the vectors. So when it is fomites, who are the fomites? Fomites are actually non-living materials or object, you can say. Okay, like let's say chair, table, any non-living objects. Okay, who can actually transmit the disease? So that's the fomites. So basically what happens in that case, like let's say I'm sneezing, okay, and all the airdrops fall onto my computer, my laptop, and then you touch, you are using and you touch, and you carry those viruses, um, and you, if you put it that entry point of that virus, then you are going to get that infection. So that's the fomites. Any non-living materials, your clothes, table, chair, book, pen, pencil, all are under fomites. Now vectors, they are the animals. Okay, they are the animals. What they do, they carry that virus or bacteria, virus from one host to another host. Okay, like that COVID-19. It came, okay, no, not COVID-19. Let's say virus like let's say dengue. Okay, dengue fever, um, or the Zika virus. I hope you have heard about that, the Zika virus. Um, West Nile virus. How you can get it? If you get the bite of mosquito, then you are going to get that one. Okay, so that's that mosquito is the vectors. So that's an animal. So vectors must be an animal who carry the virus from one host to another host. That's the vectors. Now vectors, they are again subdivided into two different type. One is called mechanical vector, and another one is called biological vector. Okay, now mechanical vector, this type of vector, what they do, they actually carry the virus outside of their body. Okay, like example, cockroaches. What they do, they carry E. coli, B. sub, streptococcus, all these bacteria on their leg, and they transmit that virus by physical contact. So what happened, that cockroach's leg has to touch your body or your food or your clothes. Okay, so that's the mechanical. Basically that virus remains outside of the vector, outside of that animal, and it's by physical contact you can get it. Biological vectors, they actually carry inside their body. Inside their body means, like I gave you that Zika virus or Zika virus, like then West Nile virus. So Zika virus, that Aedes mosquito, if you get a bite from this mosquito, then with the saliva of that mosquito, that virus, you can get that virus. Okay, so until you get that bite of that mosquito or that animal, you are not going to get the disease. Okay, so that's the biological vector. I hope you are clear about the differences between mechanical and biological. Mechanical vector, they carry virus outside of their body. Biological vector, they carry viruses inside their body. If you get the bite of biological vector, then you are going to get the disease. For mechanical vector, you can get it from the physical contact. Okay, so that's the total transmission procedure. That's how we get the viral diseases. Zoonoses, once you know the special type of diseases, viral diseases. In this case, what happened, that virus, we get it from animal host. Okay, like the virus actually originate inside a special type of animals, and from that animals we get. Okay, like this COVID-19. This virus is an example of zoonosis. Okay, because they first originated inside a bat. So they're the zoonoses. And there is another type of diseases. It's called reverse zoonosis. Means in this case, from human, any animal can get the disease. Like example is mumps. We actually, the virus, we can get the disease and we can transmit the disease to dog. So many other diseases are there. Okay, so that's the reverse zoonosis. I don't know what happens this. Hmm. Okay, maybe I have to cross it out. Okay, now the size of the, okay, so that thing is there. What happened? Okay, the size. You can understand that viruses, they are very small, because without electron microscope we cannot see them. Okay, this but there are two recent discovery, that there are Frita virus and another virus that is called Pandora virus. They are really huge, almost like a bacteria, small bacteria. Otherwise, you can see the size is mini virus 400 nanometer, yellow fever virus only 22 nanometer. Okay, so nanometer versus like streptococcus or E. coli. E. coli look 2 micrometer, much larger than all these virus. But these two virus, they are almost close to any bacteria. Wendell Stanley, he first actually crystallized that tobacco mosaic virus. Crystallized means he actually separated the liquid part and the solid part. Okay, hydrolyzation it's called. So what he did, he made a solid structure of that tobacco mosaic virus. Okay, and that's a huge discovery. We came to know the structure of any virus. That's the first one. Okay, and it helped us to isolate the influenza B virus, and ultimately we could make the flu vaccine. Until we know the structure, we cannot make the vaccine. You know, what type of proteins are there. If we don't know, and if we do not know which portion of the viral body is actually pathogenic, who gives us the disease, if we do not know that, then we cannot make the vaccine. Okay, so that crystallization process was huge, and because of his discovery, we could make that flu vaccine. The virus, they actually, you can understand that they don't have cell, plasma membrane and all those, cell wall and plasma membrane. We use very special type of stains. Stains actually goes by the alkalinity and acidity. So for the viruses, it's very difficult. So we need to make very special type of stains. We need to have electron microscope. Most of the time we do negative staining. Negative staining means we give the shape, means we cannot give the color to the virus. So what we do, we color the outside, okay, the background, and that's how we can see the shape of the viruses. Positive staining nowadays we started doing to see the internal structure. Very special techniques, shadow casting techniques. Shadow casting technique, that's actually 3D machine. We have a 3D machine. From that 3D machine, we actually make a shadow of the virus to study the virus. This is very bothering. I don't know. How can I erase this?
Yeah, it says eraser, Miss Nandi.
That's what I'm trying to do. Do you see all this writing?
Yes.
Yeah, so that's what I want to do. Let me see if I can erase this one over here. It's,
Yeah. Yeah, eraser.
yeah,
Well,
but oh
now
my
it's not
God.
highlighted anymore.
No, it's not allowing me to erase. I don't know how did I get all these things.
I don't know. It was highlighted when you switched the slides.
Huh. It's so bothering.
Maybe switch the slide, go back to the other slide.
Go back.
Yeah, and then go down to the pen, but choose eraser, not eraser pen. Now try to erase the words. Try to erase the words. Is it gonna let you?
No,
No. Still won't let you. I don't know why it won't.
no, it's not. I don't know what I did and why it's there. I really. Okay, let me go back to that annotation. Eraser. Hmm. Okay, I did it.
Yay.
Okay. Now I cannot. Um, okay. So we were here. We are done, right? This slide. Yeah, I talked about that shadow casting. Now the structure. Okay. So the general structure of virus. If we work on that, so structures of virus, it has two part. You can say that central core, that's one, and we can say it's outer covering. So these are the two main part. Okay, now the central core, inside at the center, what we can see, either we can see genetic material like either DNA or RNA. Okay, or not or, and very little amount of protein, means different types of enzyme. So virus, they're so clever. uh, they don't carry any extra luggage, extra baggage, like the minimum requirement what they need to take control over the host cell, they only carry that one, nothing else. So they carry very few amount of protein, different types of enzymes, okay, very little amount of enzymes. Most of the time they don't even use their enzyme, they use our enzymes. Like for HIV, they have reverse transcriptase. That enzyme, we don't have that enzyme, so they need that. So they carry that. Okay, so these are the two things we can find out at the center. Very few enzymes, very few proteins, and the genetic material. Outer covering, mm, just immediately outside of the DNA, they have a protein layer, solid, very structured protein layer, and that protein layer is called capsid. Now, that capsid actually is made up with small particles, small protein particles, and all those protein particles are called capsomerts. These capsomerts, they are interlinked and tightly, they are tightly linked with each other, and that's how they make a solid layer of capsid. So that's the first layer. Now, few of the viruses, not all, some of them have envelope. Okay, as I told, not all. The virus with envelope, it's very difficult to kill. Okay, the viruses who don't have envelope, they are called naked virus. The virus with envelope, they are called enveloped virus. That envelope or envelope virus, um, uh, uh, this envelope is actually made up with phospholipid and protein. Okay, these are the two main components of envelope. Now, very interesting that the virus, they use their protein. So this protein is actually viral protein. Okay, but this phospholipid, it comes from the host cell. So basically, when they make new viruses, they actually grab phospholipid from your plasma membrane. Okay, and they use that phospholipid to make their envelope. So most of the thing they actually collect from our cell. They carry very minimum luggage. So this is the main structure. Okay, now a few of the not few, actually, almost all the virus, they have a special type of glycoprotein. Glycoprotein, you can understand that it's a carbohydrate, the combination of carbohydrate, that's the glyco part, and the protein. It's a large complex compound. Glycoprotein, it's called spike. Okay, that glycoprotein. So if this is the virus, it remains like that way. Okay, so this is the tip of that organ, and it's called spike. You can say that glycoprotein spike actually. Um, this glycoprotein spike, the tip of the glycoprotein, that actually help virus to sit onto your receptor on your glycoprotein. Okay, so this glycoprotein, we actually call that as ligand. Okay, and so this is very important for the survival of the virus, the presence of spike, and this spike, or you can say that this tip part is very specific. Like let's say that H1N1 influenza, that bird flu, you know that, right? Um, that virus, they have H and N spike. So this protein part is very specific for each viruses. So that's the spike. Now, as I told that there are few viruses, they don't have envelope. They have spike, but in that case, that spike actually originates from the capsid. But for the envelope virus, that spike originates from the envelope. So basically from the outermost layer, spike originate, and it goes outward and away from that membrane. Okay. The capsid and the nucleic acid together are called the nucleocapsid. Fully formed mature virus who can give you the infection are called virion. So one virion particles means that virion is mature enough to infect you or infect any host cell. Okay, so if we see, okay, so this is the COVID virus, SARS-CoV-2 virus. This virus, it's an enveloped virus. You see that this blue structure, that's the envelope. Okay, it is RNA virus. So this is the RNA, that's their genome, and this genome is a single-stranded positive strand genome. Okay, this is the capsid. So basically what they do, so if this is the DNA or RNA, then outside they have a protein layer, that's the capsid. And if it has envelope, then outside of the capsid they have that envelope. Okay, and if they have envelope, then spike originates from here. So that's the structure. This is the central core. They have very few enzymes, and these are the outer covering. So for the COVID virus, you can see that red one, the pink one, that's the RNA. Then this part is the capsid, and small, all this structure are the capsomeres, multiple of capsomeres. Together they make that capsid layer. Outside of the capsid, they have this envelope, and these are the spikes, that glycoprotein. So that's the receptor. If it's my cell, so that's my receptor, and it will match. So virus will sit over here, and they can go through this, okay, inside host cell. So that's the science. Now the capsid means that protein, three different shape. Okay, it could be helical, it could be polyhedral, or have a complex shape. Now over here, that's the helical. COVID virus, they have helical capsid. Helical capsid, what happened, that capsomeres, they actually interlocked in such a way, they make a cylinder, long cylinder or tube-like structure. Okay, and the genome remain inside that cylinder. That's the helical capsid. Next one, the icosahedral or polyhedral, polio, rhinovirus, they have icosahedral, polyhedral. In that case, this looks like three-dimensional figure. In this case, they have, somebody joined, okay. They have 20 sides. So these are the sides, okay. And so these are the sides, and they have 12 corners. So these are the corners. And they gives a polyhedral structure, okay. So that's the polyhedral. Make sure to memorize the example of capsid. Like over here, helical, SARS virus, tobacco mosaic, influenza, measles, Ebola virus, all have helical capsid. Polio virus, rhinovirus, they have polyhedra. I already talked about it. So the difference between the envelope virus, you can see in this picture, this is the HIV virus. Okay, oh, it's written over here. So HIV virus, it has, so this is the capsid. Okay, and then outside of the capsid, and then outside they have envelope, and from the envelope you can see the spikes. Okay, all these are spikes. But this is one naked, I think, oh, this is adenovirus. Over here, you can see that this is the DNA, the genetic material. Then all these are small, these units, they are the capsomere. together they make the capsid. It is a naked virus. In this case, that spike or the glycoprotein actually originates from the capsid layer. Now the complex structure, that's the example of bacteriophage. Okay, this is the phage virus. It's called phage virus, or you can say bacteriophage. Okay, means they are the virus, they are the pathogen for the bacteria. They only attack bacteria. So for the phage virus, look, they actually look like a craft space. So a spacecraft. So what they, what they do, they actually the virus actually does not enter inside the bacteria. They only inject the DNA. Okay, so if this is the bacteria, they sit over here. Okay, they sit and they have a hollow neck. Sorry, neck, not neck, collar. They have a hollow collar, and they actually inject the DNA inside the bacterial cell. So the whole virus does not enter inside the bacteria. So over here, three main part, the head. Okay, you can say this part is the tail. Okay, tail actually head and tail are connected with the collar, and this part from collar to all the way up to this is hollow. And they have a base plate. The whole thing, this part is base plate. I think next, yeah, this is the base plate. Okay, this is called base plate, and this base plate is hexagonal structure, and at each corner, so hexagonal means like this. Okay, at each corner they have tail pins. So these are the tail pins that actually help virus to tightly sit. Okay, and these are the fibers, 6. So basically 6 tail pins are there and 6 tail fibers are there. Fibers, they're a little larger. Head of this is the capsid, and inside they carry their genome. Okay, now there's this tail part. As I told, a tail is actually a hollow cylinder. Okay, but it is actually made up with 24 annular ring. Okay, ring like structure. This and 24 rings are there. So 24 annular rings together, they make a membranous structure outside of the tail, and that is called sheath. Okay, so this sheath is actually made up with 24 annular ring, and that sheath covered the tail, original tail. So that's the structure of bacteriophage. Functions of the viral capsids, that's the most pathogenic part. Okay, like I hope you know that bacteria or any pathogen, okay, they have their special type of protein, right? That protein, once that protein goes inside, then our body, our immune system, white blood cell, they can recognize all those proteins as foreign proteins. Okay, they have capability, our white blood cells, they have capability to differentiate between the self protein and the foreign protein. Okay, so all those foreign protein, they are called antigen. Okay, so to produce the antibody, that protein or the antigen needs to go inside and has to be recognized by our immune system. In case of viruses, this mostly the capsid or the envelope, they behave like antigen of bacteria, like bacterial antigen. Exactly same way, bacteria, they produce so many different type of toxin. Their protein is different, but how that protein our body recognize, exactly same way, from the capsid this protein or from the envelope that virus protein, these two are almost same protein. Basically that virus protein, which is actually pathogenic, that gives the virus pathogenicity, that's the either the capsid or the envelope. If it is a naked virus, then pathogenicity comes from the capsid. If it is an enveloped virus, then that pathogenicity comes from the envelope. I already talked about it as inside the code they have DNA or RNA. Now, very unique characteristics. So, whole universe, all the living organism, we know that DNA is double-stranded, right? DNA, they have two strands, right? For all organism on earth, all living organism on earth, right? And all living organism on earth, they have single-stranded RNA. For the virus, they have double-stranded DNA, single-stranded DNA. They are the only organism. Okay, both type of DNA they have. They have double-stranded RNA and single-stranded RNA. So you can understand that how much powerful they are. They're much more powerful than the bacteria. They are much more powerful than us. Okay, so they have double-stranded DNA, they have single-stranded DNA, they have double-stranded RNA, they have single-stranded RNA. Horrible. Anyway, so these two are very, very important for your test. One example. So if I ask you, okay, so give me an example of organism with double-stranded DNA. So you can tell, okay, so it's me, okay, or any bacteria. If I ask you that single-stranded DNA, that's the virus. So which virus? It's parvovirus that has single-stranded DNA. Okay. Reovirus, they have double-stranded RNA. It's usually we all have single-stranded RNA. So these are the very, very important two examples. Make sure you memorize the name: parvovirus, reovirus. Okay, how viruses are classified? Okay, that's very, very important. They are so minute and they get muted so quickly. You can understand that COVID, when first entered in the US, it was so strong. Okay, people used to get high fever and so many different types of symptoms. Now nobody is getting fever, so they get mutated so quickly. All the viruses are like that. So it's very difficult to classify virus based on that standard procedure. Like for the bacteria we have that binomial, not for bacteria, for all organism we have that binomial nomenclature, right? Like one generic name, one specific name, and we based on DNA, RNA, morphology, we can classify them. Virus we cannot classify. Simple. No classification is possible. Okay, we are trying. Different scientists, different organization, we are still trying. Okay, so at this moment, based on structure, chemical composition, genetic makeup, mechanism of reproduction, by that way, so many different ways, we try it. And International Committee on Taxonomy of Viruses, they 2000, they actually couldn't classify, but what they did, they found out that there are 7 different orders, and under 7 different orders there are 96 families, and they found out 350 genera. Okay, they tried their best, okay, to try to classify, to separate out. Okay, um, so 96 families are there. Maybe now at this moment it's more, but by 2000 year it was 96 families. This were the BDD every time, and as I told that we cannot actually give them any name um, like Bacilla subtilis, so binomial nomenclature we cannot do. Okay, so families, what they used, that international committee, they used this word, the BDD, to represent family. So when we any word with this word represent the family, like parvoviridi means that's the family. Okay, and if the word ends up with virus, then that means that's the name of the virus, like parvovirus. So this is a virus. So any word with viridi, that means we are talking about the family. Any word with virus, we are talking about the specific virus. So this is the International Committee of Taxonomy of Virus. They tried this way. Another one, the Baltimore classification system. At this moment, this classification is much more popular and scientific, of course, than the previous one. They classified based on the DNA and RNA. So first they divided the virus into 2 groups. Okay, so DNA virus, RNA virus. Then they divided like DNA and RNA virus. The next category is presence of envelope, absence of envelope. So enveloped virus or non-enveloped virus. Then the next category is double-stranded or single-stranded. Okay, so they classified both the DNA and RNA virus exactly same way. I feel like it's much more scientific. Okay, so DNA virus first, with envelope, without envelope. So enveloped virus, either double-stranded or single-stranded. Non-enveloped virus, either double-stranded or single-stranded. Okay, so at this moment, we really do not have any single-stranded enveloped DNA virus. So non-enveloped DNA virus. So all the double-stranded enveloped virus, so basically all the all the enveloped virus they have, and if it is DNA, then they have double-stranded. There is no single-stranded enveloped DNA virus. Example, pox virus, herpes virus. So basically both of these virus, they have double-stranded DNA, means regular type actually, and they have envelope. Now non-enveloped, but they have DNA. In this case, so basically naked DNA virus, both type of DNA we can see, double-stranded, single-stranded. Single-stranded, remember that previous slide I talked about that the one example, that's a parvovirus. Adenovirus and papillomavirus, these two are double-stranded DNA. Now the RNA, exactly same way, enveloped or non-enveloped. Enveloped, either single-stranded or double-stranded. In non-enveloped, again single-stranded, double-stranded. First I go to non-enveloped. So double-stranded, that's the specialty, because we are talking about the RNA. RNA is almost always single-stranded. So this is the exception actually. That's the reovirus. Okay, single-stranded picornavirus, calicivirus. Enveloped virus, so there is no RNA virus with actually double-stranded RNA who has enveloped. So single-stranded genome, they are again subdivided into segmented or non-segmented. So non-segmented, these are the non-segmented example. This is the segmented. So the coronavirus, it's actually non-segmented. Very special type of um, RNA virus, okay, RNA and enveloped virus, it's called retrovirus. So this is actually for HIV, 8 patients they have. Single-stranded genome encoded reverse transcript is for HIV virus. They have a very special type of enzyme. It's called reverse transcriptase. So the word is very important, reverse transcriptase. And why it is reverse transcriptase, I will talk about it. So that's the viral classification. Do we have time still? Yeah. Okay, a little bit. Okay, now the life cycle. Okay, virus, they are a little bit moody. They have they change actually mood, frequently. Two main mood, is one is called virulent phases and another one is temperate phases. So when they are in virulent mood, you can tell. So when they are in virulent mood, what they do, they start reproducing or multiplying immediately entering up in the host cell. Okay, and then they destroy the host cell. So they enter, start multiplying, then lyse means the breaks of the plasma membrane of the host cell, and they kill the host cell, and then they come out of the host cell. Okay, so within very short period of time, everything happens, all the incident. Okay, so you get the infection, you get all the symptoms, signs and symptoms, and after certain period of time, you take medications or do not take medication, they don't care. After certain period of time, they will automatically leave your body by themselves, like flu virus. Okay, so that's a virulent phase. Within 7 days, everything is done. Okay, they will enter inside your body, they will start immediately, they will start multiplying. Okay, then after 5 days, they will leave your body. You really do not need to take any medication. Actually, you don't. There is no such medications. If you go to doctor, they will give you Tylenol kind of stuffs or cup medications to reduce the symptoms, but not to kill the viruses, because we really do not need to kill the viruses, because after 5 days they will automatically leave. So that's the virulent phase. That's special type of mood. Okay, go, attack, kill, come out. So that's the virulent phase. Another mood, this frequently they show, and that's the temperate phase. What happened? They enter inside the host cells. Okay, so if this is the host cell, and let's say this is host chromosome, what they actually do, they enter inside the host cell. Okay, and they become a integrated part of the host chromosome. Okay, and they can remain as a integral part of host chromosome for years after years. They can stay like that way. Sometimes host might not even know that he or she is carrying that viral chromosome. Okay, so they can stay like that way, and suddenly after a long period of time, or maybe within few days or few weeks, they can change their mood, and they can come out of the host cells, they start multiplying, means they can go back to that virulent phases. Okay, in that mood, they can kill, they can lyse. So I give you an example of virulent phases like flu virus over here. Temperate phase, like it's called varicella zoster virus. Okay, varicella zoster virus, you can get pox. Okay, when you are a teenagers, like within your teen years, you can get this disease pox. After a certain period of time, you will feel better, and even after blood testing, nobody can find out any presence of virus inside the body. But that varicella zoster virus might can go to temperate phase mode. Okay, what they do, they get incorporated inside your chromosome, and they can survive, they can stay like that way for 30, 40, 50 years. Okay, when you are above 60, okay, they can give you shingles. Same virus. So during this period, like let's say you got pox when you was 15 years old, and you got single when you were 60 years old. So 15 years versus 16 years. So this 45 years, what happened? This 45 years, that virus, the varicella zoster virus, was latent, or you can say dormant, inside your cell, inside your nervous system cell. Okay, there should not be any signs, symptoms, nothing, no trace. Even blood test also does not show the presence of the virus for 45 years. After 45 years, they can just suddenly then they can change their mood, and they can go back to that phase, that virulent phase, and they can start multiplying. Okay, and they can give you the disease. After a certain period of time, they will leave the cell, leave the body. Okay, so these are the two moods of virus they usually show. Okay, one is the virulent, immediate action, temperate phase. They remain inside the virus, the host cell for a long period of time, and suddenly we really are not sure what actually makes that latent dormant virus to come out, or to become active. We really don't know. We called that process is induction, means accession of you can say the viral chromosome from the host chromosome. We called when it happens, that time we called that as induction. What the reason behind induction we don't know, but if induction happens, then that latent or dormant virus become active, and then they go back to the virulent phase. Okay, so that's the characteristics of virus. I think we don't have time. I will start from here on next day. Okay, so let's keep up to this, because once I start this one, it will take a little long time. So we can end up over here today. So do you have any question? No? No question?
I don't have any questions.
Okay, so today up to this. Okay, I'm stop sharing. And we will be done on Monday, this chapter.
Have a good night.
Yeah, you too. Okay, good night, everyone.
Thank you, Ms. Nani.
You're welcome.